All right, thank you. Well, thank you very much. And you might not be, you'll be even more jealous when you see a few more photos, but we only take photos when it's sunny, right? As probably most of you can relate to. But I really appreciate the invitation to come down and talk to you under the aegis of the Water Institute. I've had a really good day talking to lots of people and hearing about what's going on here, and I'm really impressed with the breadth of people's interests who are sort of tied together under this umbrella. So I just thought I'd start with a fairly typical looking estuary from west coast of you see this sort of thing west coast of Canada or Alaska. And I was just wanted to think for a minute, what do we see when we look at a photograph like this, especially if we look at it through a scientific lens? If you're a marine biologist, you might be focusing on the estuary, which is just out here, and thinking about the mixing that would be occurring between freshwater and saltwater and what that means for ecosystem processes. And if you're a freshwater scientist, you might be thinking about the hydrology of the stream itself. And if you're a terrestrial biologist, you might be thinking about whatever it is that those people study and think about. I'm not that broad, it turns out. But we often publish in discipline in habitat-specific journals. We go to habitat-specific meetings, and we often try to stay in those zones in the colleagues that we work with. But if you look beneath the surface, you'll run into a few species like the salmon here, which kind of make a mockery of our attempt to draw lines between the ecosystems. And that is what I'm quite interested in, because the West Coast salmon have this habit of, nearly all of them die, all but one die after spawning. They're all born in freshwater, as you know. So they go from freshwater to the sea. They're out for 2 or 3 years of growth. They come back and they die. And they don't go quietly. They can die in very large numbers. They can wash up on shores of larger streams. They get hung up on logs. They can stay in the stream. A lot of them get flushed out into the estuaries. And as a result of predation by bears and wolves, of course, many of them can end up right up in the forest where they can leave a very large part of their carcasses behind. So when I see something like this, I'm sort of envisage sort of a conveyor belt of marine-derived nutrients which are coming up the streams every fall. And this goes on over a very large area. And so the area I'm interested in applying this to is our understanding of how cross-boundary nutrient subsidies work. And there are other examples. So for example, on islands, well-known examples are seaweed washing up, as you can see here, or bird colonies, where you have lots of guano deposited. These are marine-derived nutrients, and then those can wash up, those can end up on land, and where they can end up supporting some very interesting terrestrial food webs, in which a very large proportion of the productivity is driven by nutrients coming from the sea. And of course, they can go the other way. So terrestrial freshwater people are quite used to the idea that, for example, terrestrial insects fall off branches, they land in the streams, or they're gobbled up by resident fish, for example. And of course, they can bounce back out in another way through aquatic insects going into their terrestrial phase and then winding up in terrestrial food webs. So there's this trafficking of energy and material between these three so-called ecosystems, which can make it a lot more interesting to understand what's going on within each of them. And that's the topic that I'm going to be looking at today. And in the case of the salmon, one thing that's worth noting is the huge distribution of Pacific salmon. They go all the way from California and up and around to Japan and Korea. And this shows how far inland they penetrate from the various species that are there. So if there is something going on, it could be affecting a fairly large area of freshwater and terrestrial real estate. So this person's already got it figured out. He is holding in his hands, his gloved hands, a frozen dead salmon. And he's about to, do you know what he's about to do with this salmon? Right? What's he going to do with this salmon? He's going to chuck it into the stream, right? He's going to throw it into a stream. He's got a whole truckload of them here ready to go. So this is a dead salmon. They've trucked it up here. It's come out of somebody's freezer, and they're going to throw it into the stream because they think this is a good thing to do because the nutrients then are going to go and help sort of kickstart the ecosystem and perhaps create more salmon in future generations and so on. You can even find a book, a little guideline that the Department of Fisheries and Oceans wrote on salmon carcass planting procedures for Vancouver Island. I don't know why Vancouver Island has a different handbook from other places, or maybe they just didn't get around to it. But there's already this idea that these carcasses could be important in sustaining freshwater and riparian ecosystems. But there's a bit more to the story. If any of you have ever watched salmon spawning, here's a video I took a few years ago up in our study area. Let's just think about what's happening. These are chum salmon. So here are some, you'll see, here's some males fighting, jockeying for position. You can see they're not subtle, and they're smashing the rocks around. The females are digging nests with their tails, smashing things around. They're going to be suspending a lot of particulate matter. They're going to be clearing off and scouring the area. You can see there's a bit of a mosaic here of areas with algae on the rocks and areas without algae. And so there's a whole lot of perturbation and engineering going on as a result of their spawning activities. And so some people have actually argued that they might actually be exporting more nutrients than they're bringing in, because they're actually sending these things downstream, and in a short coastal stream they might be sending them straight out of the system. So it's not as simple as the person with the dead carcass might think. They're having some sort of an effect, but we're certainly not mimicking what actually goes on in wild streams. Here's another way to look at it. This is a photograph from the Skeena River in northern British Columbia. It's a very, very large and complicated watershed that it's part of here. And these here are basically gravel bars that have been created as a result of the spawning behavior of Chinook salmon. So Chinook salmon can be very big. It's the biggest salmon. And they can move very large boulders when they're digging. And then these are accumulating. They're basically these are the result of their actual spawning behavior. They roll downstream and they're accumulating in these patterns. So when we say ecosystem engineer, that's probably an apt description here again, just to emphasize that they're having more than just a nutrient input effect on the streams. So I wanted to ask, what are the ecosystem effects of salmon? And toward the end, I'll just take maybe 3 slides to sort of with some thoughts, and I don't have answers. It really is a question that stays a question, but some thoughts about what we might do with this information if we're interested in ecosystem-based management. How could we incorporate this information into what we might be trying to accomplish in terms of conservation of salmon as well as their wider ecosystems? So we're set up in an area called the Great Bear Rainforest. This, you can see it's a big area here from just up, well, sort of northern, adjacent to northern Vancouver Island all the way up to the Alaskan border. And you see how green it is? This is a map of logging that has occurred in BC since 1970. So it gives you some idea that it's a little different from everywhere else in southern and middle BC, in that it hasn't been logged nearly as much as many other areas. Some, even in Haida Gwaii here, which we think of often as, for its wonderful rainforest. In fact, it's been clobbered very, very hard before some contentious agreements finally led to protection of some of the regions there. But the thing here is that there's no roads into this zone here. There are no roads at all. And so it's very difficult to get to, and the loggers didn't get there, except for some selective high grading in some of the coastal river valleys. So it's a good place to look if you want to go to somewhere where there might be somewhat semi-natural ecosystems. This is not to say it's never been impacted by people. It's been heavily impacted by the fact that there are 10 to 11,000 years of continuous occupation by First Nations people there. For sure, if you have your eye in, you go out with an archaeologist, they'll show you the middens, they'll show you the campsites, the fish traps, and so on. So I'm not saying it's not he'd been impacted, but people have been part of these ecosystems for a very long time, but not in the way that most people are today in terms of land conversion. And so we work. I'm based on this is Bella Bella. I'll show you a photo of Bella Bella. That's on one island. We're based, and that's the First Nations village for the Heiltsuk people. We're based on this island over here in a place called Shearwater. It's got there's an old cannery where we're set up with an apartment there. And these are all places, these are all streams that we have visited at one time or another during our research. And for scale, this is about as far as you can get to in about 2 hours of boat time to get up there. So all these sites are reached by boats. And we've done various amounts of work in a number of these watersheds. And so here's Bella Bella. That's the island. That's about a 15-minute boat ride away from where we are. And we're working with people from the HealthSec Integrated Resource Management Department who have a fisheries office. And we count salmon with them. We give them all of the data at the end of each year. And we've been involved with salmon camps and various other activities in the local schools there. And we also have a number of people every year working with us. And basically, their job is to provide adult supervision to my graduate students. They know the area. They've been very much involved with our research over the years, and we're very grateful for the privilege of being allowed to work in their traditional territory. So here's what my office looks like, and rare times when I'm actually in the field with my students. You know how it is, right? I sort of go up, disrupt them for a little while, and I leave. This is one of the very nice fjords. There are mountain goats up on the tops of these slopes here. Huge numbers of streams, which is why I chose it. I wanted a large number of streams because the unit of replication for nearly everything I'm going to talk about with you is the stream. And every stream is straight into the sea, so it's essentially a watershed. A huge variety of watersheds, and that we can make comparisons for looking at the effects of these fish. I'll introduce the players, chum salmon and pink salmon. We don't have sockeye in the streams that we work on. There are some sockeye salmon around, but I'm going to be talking almost entirely about pink and chum salmon. And we have coho salmon in all of the streams, but they tend to spawn further upstream than where we work, and their carcasses are not really part of the story. They don't deposit a lot of carcasses that find their way to where we work, but their juveniles will be part of the story because they're juvenile coho, the little ones that spend a year in fresh water. They're there all the time, and so we will be talking a bit about those. The other players are the bears. We have both bear species and these fishing wolves. This photo was actually not taken in our Stouty area, but we have these extremely marine wolves, a very high percentage. The majority of their diet is marine things, and they're very good at fishing. As you can see, they're not scavengers. These are fishing wolves, and they account for a very large proportion of the carcasses that get deposited on the stream banks, especially down near the salt marshes, near the areas where they keep their pups and so on. So we have both, and actually this area has arguably the most intact large carnivore assemblage in North America. Even in the Arctic, which those who haven't been there tend to think of as reasonably pristine, but as we all know, the Arctic is very much used by people, and in terms of with the overall lack of hunting going on here, except for a very controversial trophy hunt for grizzly bears, these carnivore assemblages are basically pretty much as they would have always been. And that's important because they're the ones that drag a lot of the carcasses out and leave them there for me to study. So here's my boot and a chum salmon. Now that's been taken by a wolf rather. They tend to eat the brain or the head, and they tend not to eat the musculature. It's thought that's because there may be a parasite in the body musculature that they can get. It's not for sure. We don't know, but most carcasses you can tell who did it. Whereas a bear will actually eat the body muscles and gonads and so on. So I'm going to just give you a fly through some highlights of some of the areas that we've been studying. I'm going to first of all start with some freshwater examples. I won't talk about every link in this sort of simplified food chain. I won't talk about periphyton, but we've worked on that. We'll talk about aquatic stream insects, coho, salmon. I won't talk about our sculpin work, but just to give you an idea of the way things are sort of put together. And then we'll also spend a little bit of time on land. We are doing some work on bears. I won't mention that, but we will go into plants, scavenging birds, and breeding songbirds. There is a marine axis here too, which I'm not going to go into. We've looked at estuary productivity as well. But what I'm going to do is I'm just going to give you some highlights of some of the work we've been doing, asking this question of whether there were detectable impacts of salmon on various elements of these ecosystems. So I want to begin then with work on aquatic insects. So we've had a couple of people working on this. And actually, don't just work in we work in the Fraser too. But I'm going to talk about Jennifer Harding's work briefly for her PhD. And she was studying both periphyton, which I'll leave out, and also stream insects, and simply asking what the effects are of salmon on the insects in these streams. So here's the N15, sort of stable isotope, heavy isotope of nitrogen. ratio. And here it is plotted against salmon density in the spring and the fall. And so what we have here, I've actually lost my legend, but I'll make it up. Here's the density of salmon. So each of these is a different stream, although we may have more than one trophic group of insect along the way. The top line are predators, okay? The bottom line are grazers. And so basically there's this tendency for all of the groups, especially the upper levels, to increase as in streams, at least their N15, sorry, the N15 trophic signal is increasing because salmon have a very high N15 signature. And that's particularly true in the fall. And that, of course, is when the salmon are there. But we can still kind of see a bit of a legacy of this in the spring invertebrates that are there as well. So from that, you might think, okay, there's a benefit somehow to the insects. They seem to be consuming salmon-derived nutrients. We think a lot of this is coming from the algae, some of and the periphyton that it's scraping. Some of it may be coming directly from the carcasses themselves. But if you ask about the actual biomass of invertebrates, which is what I'm a lot more interested in fact, it's the opposite. So here we see different streams in the dots here, the density of these streams from very few to lots. And you can see that in the spring and the fall, we get actually fewer invertebrates in these streams when there are more salmon. We've had, this has been foreshadowed by some work that was also done in Alaska, and I think that this is the scouring effect that I was referring to. I think that the salmon, when the salmon arrive, they simply cause a drop, a very immediate drop in the spawning reach in which the salmon are there, and I think it's because of this scouring effect. So this is an example of the engineering effect. Although they're taking up the nutrients, I think the engineering effect is having an overall cause for an immediate drop in their numbers. Clearly those numbers are rebounding by the time the next year comes around, or else there'd be no bugs left. But it does appear that they're causing this localized decline. So now I want to talk about fish. And as I said, we've studied two species of sculpins as well as coho. In the middle here is Michelle Nelson, who did her PhD on these. And the thinking behind this one was that this is the chum salmon with those sort of wine stains down the side. The chum salmon does not spend any time in fresh water after they hatch. So they spawn in the fall, the eggs go in the gravel, they overwinter, out come the fry in the spring, and they go straight to the sea. When they're really tiny, they're straight out of the egg. They use up their yolk sac and they're gone, okay? And so they have no way of benefiting from salmon-derived nutrients from generations past because they don't feed in fresh water. Okay? But coho juveniles do, because most of our coho spend 1 year in fresh water, and about 10-15% spend 2 years in fresh water before they go out to sea. So the question was whether chum and pink salmon, which have the same life history, straight to sea, whether there could be their nutrients could be crossing over through the food webs and affecting the growth or survival and densities of juvenile coho salmon. And so we've done all sorts of methods. We started off with just simple minnow trapping, which eventually turned into pole seining, and it turned into electrofishing. And we've put things together in various ways and tried to overcome various biases here. And here's an index of juvenile coho salmon. So every dot here represents a different stream, and the abundance of, in this case, of pink salmon. Pink salmon turns out, as you'll see, to be the best correlate of the abundance. And you can see if you have few or in some cases no salmon because of waterfalls right at the mouth of the stream. So we have some ultra controls where there's no salmon at all. And you can see it's quite scattered at low numbers, and then it runs up to some sort of an asymptote here in terms of the abundance of these juvenile coho salmon. And here's what it looks like if you compare different variables that might explain the summer coho. Pink salmon and stream size together are in the top model with not a terribly high model weight. In other words, the probability that that's correct. It doesn't really win out in a big way, although the R-squared is not bad. Or pink salmon by itself, or chum and stream size. So the trend is that out of 17 habitat variables that were predicted, the top models have salmon in them and stream size. And in fall coho, it's a little bit more of a certainty. Again, it tends to be pink and stream size that win. Now, this we believe, is not just because streams that are good for pink salmon and chum salmon are also good for coho. Right? You could argue that that's just a spurious correlation, not a causative one. And it is a correlation, so we can't be sure that it's causal. But we have analyzed these 17 variables every way we can, not only to try to explain the juvenile cohos, but also to understand the numbers of chum and pink salmon, the nutrient producers. And the long story short is that this is the most parsimonious explanation, is that this is not a spurious correlation. The correlations are there, but they're not so high that we're breaking any rules by putting them both into the same model and so on. So we think that this is probably the reason it happens, but we haven't done an experimental, it'd be nice if you could take a whole stream and keep the fish out one year and see what happens, but nobody's going to let us do that. So then I wanted to now move on to land a little bit and talk a little bit about the bird work. And this is work that was kicked off by Rachel Field. We've pushed this on a little bit since her time there. I'm an avid birder, so it was, of course, only a matter of time before I tried to figure out whether we could try to tie salmon to bird populations. And my original idea was that I would do point counts along streams where you stop and listen and, to all the birds you can hear. The problem is when you're standing in a stream, it's very hard to hear the birds. And so I thought the easiest thing to start with would be to simply look at the forest birds that are breeding around estuaries, because nearly all of the detections here are just by ear. And so that was Rachel's job for 2 years. And we did it both in the fall, counting the scavenger birds, and we also looked at the songbirds in the spring. So here's an example of some fall research. So here you have the total, again, carcass biomass. This is almost always on my axis in some sort of units here, and an index of scavenger abundance. And this includes ravens, crows, about 6 species of gulls. There's about 4 common ones there, bald eagles, and so on. Now, streams are represented there more than once here. We didn't have that many streams. So in some cases, this represents multiple visits through the fall of the same stream. But in the statistics, we accounted for that. And you can see there's a reasonably strong relationship here. Still lots of scatter between the numbers of scavengers that you see in the fall and the biomass of the carcasses that are coming down from the streams here. And I don't think that's the most earth-shattering discovery, and I think Rachel would agree with me. Basically the birds are distributing themselves according to the abundance of their food, and they're moving among them. I, of course, what we're now doing, I said, I'm being somewhat mischievous. I said, if we were to we could flip these axes now and use the birds to predict the number of salmon in a stream instead of having to get out of the boat and avoid getting eaten by bears and having the boat drift away because you didn't anchor it properly, and et cetera. Because this takes about 15 minutes, right? So we're now working on some simulations to ask how much more information, how many more streams could you look at in a cost-benefit framework compared to the loss of information by not counting the fish. I don't expect anybody's going to we'll never get it published because people have their ways of counting fish. But anyway, we're just going to play around with that a little bit more. One thing that was kind of cool was that we also asked whether it mattered. We looked at all the different species. They all kind of came out the same way. So you don't have to distinguish among gulls. Not everybody can tell the gulls apart. That doesn't matter. You don't have to distinguish among the gulls because they're similar. The one thing was that if you compared senescent fish that had just died after spawning, or ones that have been killed by predators, eagles were most correlated with freshly killed carcasses that have been opened up by predators. So the eagles obviously are more interested in the fresh prey. It's not that they can't open a carcass. It is hard actually. For the smallest gull species cannot actually easily puncture and rip into carcasses. They tend to take eggs. But it does seem that the eagles obviously could rip open a carcass if they want to. But eagles seem to be associated indirectly, therefore, with large carnivores that are making these prey available to them. That was new. Then we asked, what if you go in the spring when the breeding birds are there? So this shows the density of birds. So it's controlling for the size of the estuary. It's per hectare, okay, against, again, pink and chum biomass together in streams. And each stream is represented in 2 different years. You can see that if you look on the far side, right? There's 2 different years there. We had fairly consistent results between the streams. So a stream with a lot of salmon over here, has a lot of salmon the next year, and the bird and the birds are fairly similar in that stream, this one in here, and so on. And again, as seems to often be the case, it's highly variable if you have very few or no salmon here, but then it seems to go up to some sort of an increase here in the number of breeding birds. Now we've looked at all sorts of additional covariates, because we always have to with these correlational studies. That's why I needed big sample sizes as much as we could with these streams. And this seems to be the most parsimonious explanation. But I want to point something out to you. This Townsend's warbler and this Pacific slope flycatcher, which are a fairly prominent part of the data set, they've never seen a salmon. Okay? They've never seen a salmon carcass, they've never seen a salmon egg, because they're migratory. Okay? They're gone by the time the salmon arrive in the fall. Okay? So this is not any kind, for them at least, it's not because there's a direct, they're not feeding on eggs, they're not feeding on carcasses or anything like that. Now these other ones, we occasionally, like I've seen varied thrushes certainly picking at salmon eggs. I've never seen a kinglet. Kinglets are insectivores, they're not going to be doing it. But the point is that these breeding birds, assemblages, are occurring when there are no salmon in town whatsoever. And so what we think is probably happening is that the overall productivity of the estuary may be enhanced by these carcasses coming down, and perhaps it's through the insect prey. It's also possible, and we've tested for this, that it has to do with structure of the vegetation being somehow affected. And so if you look at the total density of birds, for example, the top model has salmon and estuary area, even though it's a density estimate. So even though we've sort of controlled for it, there's still something about the larger estuaries. I guess there perhaps is additional productivity and so on over and above just the size of the strip of land around the estuary that we censused. Or it could be salmon, estuary area, and shrubs. So we did all sorts of forest plots here. And again, similar idea. I guess my point is that salmon, although they're appearing in all of the models, I don't want you to think it's just salmon, and I don't want you to think salmon are only, they seem to be, if you put this all together and average across the models, they are the top model, they are the top parameter, but it's not just salmon, and it rarely is just salmon. It tends to be salmon along with some of the other things that you might expect. And this is one, for example. This is forest height diversity. We've known since about the 50s from Robert MacArthur's classic work that forest height diversity is related to songbird abundance. It's a classic measurement. Ornithologists use it all the time. It just has to do with looking at the different layers in the canopy and calculating the overall diversity of the canopy. So this sort of makes sense that it would be, in these models as well as the salmon. And so both are in these top models here. So it's not, that's for diversity. So it's not just salmon. I guess that's my point. I think it is certainly partly salmon, but not by themselves. So then we were also quite interested in plants. So the first big push on this study was in 2007. And so we got a crew of people together many people were doing different things all at once here. And we decided to try to do some plant surveys to see if these might be involved with plants. First, I want to show you the only experiment that I'm going to mention during this talk. This is one of our study areas, Ripley Falls it's called. It is a big waterfall. It's right at the mouth of the river, and no salmon can jump this thing. There's no salmon habitat, as you can see down here, okay? So, and you can see it's a pretty substantial thing. So, Morgan Hawking, my postdoc, and I got the bright idea that we should put some salmon up above it and see what happens to the plants. And so we went to a stream, not even all that close, some distance away. We boated over to this other stream, got a whole bunch of rotten carcasses out of the water. So these were truly disgusting carcasses because we thought they'd be less likely to be scavenged. We put them in dripping plastic bags and then hiked up over through the bush here over this waterfall together, right? And this was the point where Morgan stopped and said, "Geez, John, I hope we don't win a Darwin Award for this," right? Because we got these dead salmon in grizzly bear country, and they're big, it was heavy, and so we did this there, and we did it in 10 more streams. And, and we laid out a series of carcasses, usually about 10 per stream. And here's how it went. You'd stick down a carcass and flag it, and then, and this is the kind of thing we were manipulating, and then we would take a sample of up to three, if we could find them, 3 species of plants, which I'll call on. So we take 3 species of plants right here, foam flower, bunchberry, and false lily of the valley. And also lanky moss, which is the dominant moss that carpets these areas. And so then we'd put down the carcass, and then we'd go 2 meters away, and we do take another sample of the same species. So we have this is our before, obviously, right? So that's our before. We have the salmon. These should not be different from each other because we just put the salmon down. You go back in the spring, in fact, these had a total of 8 months to sit there. Oh, I thought I had a photograph of it. And when you come back, you find just the skeleton, and nearly always the carcasses were still there, and you could tell they'd been there a long time because there's sort of a burn area around them from the effect. So we'd go back in the spring and we do it again, right? Sample them again after they'd gone through the fall, winter, and a good bite of the spring. And here's just an example of the sort of results we had for these 3 herbaceous plants, percent nitrogen in their leaves. So in the fall, the on and off was not different, and it shouldn't have been because the carcass hadn't worked its magic yet. And then in the spring, there's this bounce in the percent nitrogen in their leaves. There's a slight increase off the carcass too. Perhaps that's just a spring growth effect, right? But with this before-after control impact thing, we get a pretty clear signal here that the plants are in fact showing a nitrogen response as a result of having had a salmon carcass placed beside them, which is kind of interesting because we actually, some botanists that we'd spoken to were doubtful that we would have such an effect because they thought, because it wasn't really most of this was not really happening much through the growing season. It was, much of this was through the winter, but there was some time certainly in the spring for this growth effect to happen. So the percent nitrogen went up. We had interestingly kind of variable effects on the N15 signatures. Most of them went up. They actually went both directions with our mosses, and we think we know why that was. But it at least is some sort of evidence that these plants might be taking up these nutrients and incorporating them into their leaves. And here's an example. This is the lanky moss that I mentioned, a very common moss, and lily of the valley. And so here's salmon stream density here, and again, it's sort of the on and off. So the on, that's this effect that, you know, of higher percent N, and you can see that if you have very few salmon in the stream, there's a bigger difference. There tends to be a bigger difference. So these are the least nitrogen saturated. Here, it's not having quite as much of a difference, at least for this species. It's interesting, it is still having an effect on moss. I'm kind of surprised by that. But it suggests that in the really nutrient-starved things, you might have more of an effect, whereas these other ones that typically have lots of salmon of their own coming, so I showed you the control where there'd never been a salmon. We had lots of streams where there were salmon, and they tend to differ somewhat in their responses. So what I really cared about, though, was the actual species that were there, the species composition of the riparian plants. And so we threw some quadrats. We went to 50 streams, and we ran a transect 35 meters up the stream, 5 of them on each stream. No, 6 of them. So yeah, 6 of them, 3 on each side. 35 meters up, we throw down the quadrat. We quantified the percent cover of mosses, the herb layer, and the shrub layer. And we also measured the diameter at breast height of over 6,000 trees along the tape, 5 meters each side of the tape. So we had the forest plot, sort of tree cover. This is Morgan here. So we had the tree cover as well as the plant cover, and we crunched this all together. We ended up with over 30,000 data entry points just from the quadrat data alone from across these 50 streams. And so we're going to ask, as usual, how these might be related to the salmon. And we were able to predict beforehand which species might respond because there's a handbook of forest indicator plants of BC. And this was done for forestry reasons, and it's simply because you could then diagnose productivity of the forest by looking at the species of plants. It had many, many things other than just nitrogen indicators. But from that, we had this independent, a priori predictor where we were able to array the species on a scale of how much there would be nitrogen indicators or not. It's actually a continuous scale, but I'm giving you two examples here of the extremes. So lily of the valley, salmonberry, stink currant are examples of nitrogen-rich indicators, whereas things like salal, this is a shrub that you find growing anywhere, in the sidewalks beside, in Vancouver. It doesn't require large nutrient inputs. And so that gave us some prediction of who should benefit and who should not. So let's take a look at just some examples from this. So here I'm just showing you two of the nitrogen poor indicators. These are the nitrogen rich indicators. Here's the salmon stream density, and each dot is a different stream. Okay, so one stream, and here's the N15 signature. You can see that actually they're very similar. They all tend to increase if there's more salmon. Okay, even in the N poor indicators are showing a higher N15 signature. If you look at the percent N, though, only these two species are showing a response. These are all over the place. The N poor species. They're not obviously. It's not obviously causing an increase in the overall nitrogen in their leaves. And then if you look at their relative density, in fact, these tend to come down, and nothing there. Salmonberry can become quite dominant in streams that have a lot of salmon. So it's a well-named plant. The salmonberry does indeed like salmon. And so these are just, I've chosen these, well, we chose these examples to illustrate, but I can also show you this on a broader sort of community-wide scale. If you take all the species together and array them on an axis that, of the N-rich community, so this includes all of the species going into an axis that distinguishes these two communities, there's the overall result with stream salmon density, and each of these is a different stream, and you can see that you get these species dominated by false lily of the valley, salal, stink currant, things like that, whereas things like deer fern and false azalea and other things like that tend to be at these low salmon streams. It's the first time someone's shown a response of riparian vegetation that matches salmon. Another way to look at it is the distance from the stream, because we actually threw our quadrats at varying distance. And so if you divide the streams into low, medium, and high numbers of salmon, this is simply showing the same thing. It flips toward the nitrogen-rich community. And if you look at the distance from the stream, it's especially clear close to the stream bank. And the further you go from the stream bank, up to 35 meters, the less of an effect it has. So it's attenuating over this effect is attenuating over the 35 meters that we did our sampling. Okay, it's definitely attenuating. So it's interesting. It's not a big, it doesn't go a long way. From other work we've done, these salmon nutrients are probably not, in general, going detectably more than about 50 to 100 meters off the stream. And the interesting thing is that there are some really neat interactions that shape the responses, because everything I showed you in those graphs showed an awful lot of scatter, right? A lot of scatter. And so we looked at whether things are alder-dominated or cedar-dominated. So this is the slope of the relationship that you get in the stream community in alder-dominated versus cedar-dominated streams against streams' salmon density. What this is saying is that whether you have very few salmon or a ton of salmon makes no difference to the uptake, to the community, if you have a lot of alders. And that makes sense because alders, of course, are nitrogen fixers. So that these systems are not as salmon nutrient dependent because there's an alternative source of nitrogen from the alders. And I'll just mention the steep banks. Actually, I'll do that one. Let me just do it with a photo. I think that's the next shot. Steepness of the banks matters. If you have really steep banks here, you get no detectable effect of the salmon on the community of plants. And it makes sense because the bears are not going to come down these banks to hunt. And then if they did leave a carcass lying around, the carcass won't stay there. And so if you have steep banks, that doesn't have much of an effect. And this is just that alder effect. These kinds of streams are basically insensitive to what we're getting from these nutrients. So, as I mentioned to a couple of people this morning, I sort of felt once Morgan Hawking, the postdoc who led this research, once he started showing me those analyses, I started to actually think we might have it, because you always worry about a correlative study, but in some senses these are exceptions that help, I think, prove the rule that there are some sensible correlates of the overall effect, and the effect goes away in certain types of streams where you would actually predict that it might go away. And then the last thing I want to say about plants is a recent project that we did with, led by Leslie Herteau, and this was also in collaboration with Morgan Hawking and Arna Moores in our department. And we looked at phylogenetic filtering. So the idea here is that if you have very few salmon, there may be only certain types of plants that are able to thrive there. And in particular, these would be species that have mycorrhizal relationships with their roots and which enable them to take up inorganic forms of nitrogen. And on the other hand, if you have a lot of salmon, that might leave room for other species to potentially come into play, species that are not necessarily closely related to each other. These might all tend to be ericaceous shrubs, for example, which have those characteristics, whereas species, you could have a much more dispersed phylogenetic tree. So the idea would be that these, a lack of salmon could cause a filtering of your phylogeny in a more clumped phylogenetic tree. So we put together a phylogenetic tree of about 65 species of vascular plants from our analysis, and here it is. So here's the salmon density. Each dot is a different stream, and this shows you how clustered or dispersed the phylogenetic distribution of the species in the community is. And that is the effect we get, that we get a greater dispersion at high numbers. You can see it's not exactly a monotonic. I mean, like the scatter is enormous here, and perhaps that's not surprising in light of what we've learned about what controls these plant communities. But once we get out here, it's a fairly consistently more dispersed phylogenetic assemblage of plants on these streams, even though the overall diversity the overall actually species diversity can go down. So I'll just summarize some of these highlights and before we think a little bit about what we could do with this if we want to go into the next step, which we haven't taken yet, but I am interested in doing. Here you can see, I haven't mentioned this, we get variable effects. They do actually scour out the phytoplankton, though it takes up nutrients. Variable effects here, we saw it takes up the nutrients. These bugs take up nutrients, but they can actually decrease. And in fact, we've done this research in the Fraser River and found in that study that they actually can increase. So we're not quite sure what the bottom line is for insects. We saw positive effects on relationships, at least with the density of these, and we've also found it with growth. And I haven't mentioned sculpins, but they had the same response. And on land, we've seen all sorts of correlates here with plants, scavengers, and songbirds. What would you do with this if we wanted to look at ecosystem-based management? Here's Canada's wild salmon policy. It was published in 2005. This thing probably had one of the most painful gestations of any document ever produced by government. It took them nearly 10 years of public consultations to say, what are salmon for? What do we want to do with salmon management? And this involved lots of meetings. This is all before my time on the West Coast. I was living in England at the time. And they had all sorts of meetings with First Nations people, various stakeholders such as fishermen and anglers and members of the public. And it's a very good document. I think sets out a very nice blueprint for the objectives of managing West Coast salmon. And it says the challenge is a need for development of an ecosystem objective that is widely appreciated but difficult to quantify because it acknowledges that we should probably not just be managing salmon for our own fisheries, whether aboriginal, commercial, or recreational, but also for these wider ecosystem objectives, which 10 years ago we kind of knew they were important. We knew that grizzly bears ate salmon, of course, but as it says, it was very difficult to quantify. Well, I've spent some time now quantifying them. I've shown you lots of quantifications of relationships between salmon and various potential ecosystem objectives, okay? And it said few studies at the time provided the advice on the number of salmon necessary for healthy ecosystems. There was a very clever document. They didn't try to define healthy. It's a good thing. That could have been another 10 years. We know one when we see one, right? Whatever it means, it doesn't matter. The point is the objective was there as a blueprint for an aspiration. I wouldn't have tried to identify what that means exactly either. So let's see what that might look like if you're going to bring that into the world of fisheries management, which is the way we currently are doing business. So let's just look at a typical yield curve here. This could be for any fishery. If you have the number of salmon going from 0 to maximum. So this implies that there's no catch. Okay, this implies that there's no catch, so they're all getting back and spawning, and this implies that you're catching them all, and most of the fisheries are happening at sea, and so certainly they're happening before the spawning grounds. So, and if you do that, you can see there's obviously a trade-off. If you let them all get back, your yield to the fishery is 0, and and if there aren't any, you're not going to get a yield either. And this is our classic maximum sustainable yield trade-off. And in this particular case, if the curve is shaped like this, this would be what you would shoot for. That would be your target if you're trying to maximize the yield to the fishery. Okay. Now, what would a ecosystem look like? What would a bear's eye view of this be? Well, if there's no salmon, that's not very much of a yield to the ecosystem around the spawning ground either. And it may well be, though, that the ecosystem yield is going to be maximized way out here, not where we maximize it. Okay? This is our zone of conflict here, right? If we're going to decide are the fish for us or are they there for the bears. So from a bear's eye view, your EBM, your ecosystem-based management target would be here. Okay? And this is our zone between the two extremes. That's our zone where we have to make a decision. We have to decide on a trade-off whether we mean it, that we're really going to give up some fish for these other objectives. Now, if bears were the only thing we cared about, we could do this right now because this work has been done, partly by researchers in Alaska, partly by Chris Darramont and colleagues at UVic. You can do it. We can do this for bears pretty easily. But is that what we care about? What if another species has a different looking yield curve? I mean, maybe it would, like, if this were what eagles looked like, well, then they have much less of a conflict. So if we wanted to maximize for eagles, we might have less of a trade-off, but then we're not doing the bears any favors because we're here. So you see, the problem is that I've just shown you probably 12 or so, maybe 15 of these curves, exactly these curves, where numbers of salmon versus effect of some sort on an ecosystem. And the question is, where do they all end up? Do they end up in the same place? Well, I'm not the only one doing this. Tom Reimken's group, for example, a number of years ago, looked at carabid ground beetles and pitfall traps beside some streams, and here's the N15 signature in those and the salmon per meter squared. Now, an ecosystem objective, I know that the public probably isn't deeply concerned that the carabids have a high N15. That might not be quite the target, but maybe it's an indicator. Maybe it's an indicator, not necessarily the target, but it's an indicator of that kind of a target. So we went through the literature and we looked at all of our cases, all the other ones. We went through some very painful standardization exercises of trying to get this axis to be the same, because even in my talk I've shown you several different metrics for this. And if you put it all together, this is what we get if you wanted to look for maximum ecological impact. So that basically, where's the asymptote on those curves? Some don't have an asymptote, okay? So if you take the examples of N15 signals in all kinds of things, okay, bugs, plants, and so on, you can see that here are the number of studies that show that you can actually maximize it just above 0. That doesn't mean literally 0, but just above 0. So there's 6 in that category and 3 more here and a couple more at 5. In other words, it seems like the tracers these indicators tend to max out very soon, very smallish numbers of salmon. So I'm not actually sure, unfortunately, that this is a great way that you could diagnose the health of your stream. You can't just take the leaves off the salmon berry or what have you, because they tend to max out fairly quickly. If you take some studies of birds, we've been pushing this a lot further now. I have 2 more graduate students working on birds who I haven't told you about, but those are just 2 examples of studies. There's one on periphyton, actually, and there's a bear study, and so on. The thing is that they're all over the map, okay? And this is partly, I think, because they're different species that have different biology. It's partly because different streams have different capacities for how many salmon they could support anyway. There is no necessarily one metric of density. And so I think where we are now, and this is actually an area that I'm now starting to get into, is to think carefully about the scale of the management and the trade-offs that would be involved under different scenarios. And so I think my question, I began by asking the question, what were the ecosystem impacts of salmon? Well, there are clearly quite a lot of them, and I think they could translate into management if people decide on both the targets and the trade-offs to tie these together. And that's very much what a number of agencies are now saying that we need to be doing, and I'm hoping that that'll perhaps be sort of phase 2 of our research. I think the ecology was kind of the easy part, although it didn't seem so when I was carrying a transect tape up a muddy bank in the pouring rain. But now we also need very much to move more toward the management side and including people in these equations. So I just want to mention, because I hope it was pretty clear, that an awful lot of people were involved in this study. This is a snapshot in time. There have been other graduate students coming and going through this program. I particularly wanted to acknowledge the Healthsick First Nation, who have allowed us to work in their territory and to work with them. This has been in many ways the most satisfying aspect of our research there. And of course, I wanted to thank you and the Water Institute for bringing me here. So, thank you.